Hey there, it's Mrs. Scoville. I'm a little bit gravelly voiced today, but we're going to be learning about Mendelian inheritance. We are in our genetics unit. There are two objectives for today's lesson. The first is to identify a phenotype when given a genotype, and also to write a genotype from the terms homozygous, heterozygous, and dominant and recessive. So we're going to start out today's lesson a little bit differently. I want you to take just a minute and think through this. I have two questions for you. We've got these two rabbits. One is black and one is white. In your notes, write down what color children you think these two rabbits would have and why. And if they had a lot of babies, do you think all their baby bunnies would look the same? Why or why not? So just take a minute. You can pause the screencast here and write down what you're thinking. Okay, so the next part, what we're going to be doing is looking at the work of Gregor Mendel. He was a monk who worked with pea plants to learn how genetic material and traits were inherited. You're going to watch this video, which is linked in your notes, and you need to write down 4 observations after you watch the video. So again, at this point, you'll pause. Gregor Mendel, through his work with pea plants, found kind of this gene term. And Mendel, when he was doing his observations on the pea plants, saw that there were certain factors that were passed from one, what we call parental generation, to another offspring. So this was his conclusion from watching the pea plants, and especially looking at the wrinkled and round and purple and white traits. We know today that this is through DNA sequences. Mendel didn't discover DNA, but we know genes are on DNA. So again, gene, that talks about the factors that are passed from one to the next. There are different versions of a gene, though, just like white and purple for flowers, and these we're going to call alleles. So we can think of this again. Remember, genes are on chromosomes, which are made up of DNA. And if we're looking at a specific gene on our chromosome, 1A, this one could be a purple. We're going to have a different phenotype for the same thing in the same location. Those would be alleles. Okay, they're going to have different, and we'll get into phenotype in a second, but different versions. Okay, so genotype and phenotype. Genotypes refer strictly to the genes and the gene combinations you find in an organism. Phenotype is what we see externally, so pH, physical. It's the physical traits you see in an organism. So that would be, for example, like in this picture, your genotype. It's the chromosomes with the individual genes on it. These are going to encode for what you see in a phenotype. And for this mouse, that might be things like the brown color, or even like how long its whiskers are, how long its tail is. When we get into looking at alleles, though, Mendel came up with a second conclusion, okay, and this is known as the principle of dominance. And what this told him was that there are going to be some alleles that are dominant, so that means when they're there, that's the phenotype you see. And we know, if you look back, we have 2 copies of each chromosome, as we've seen multiple times, but when that dominant one is there, then it's going to be expressed. So dominants are always written as capital letters, so an example of this would be like a capital B, okay, maybe for brown in that mouse. Some alleles, though, are going to be what's called recessive, and recessive traits are only seen if you have 2 copies present and there's no dominant there. So as long as there's not a dominant gene, then you can see the recessive. This usually means you have to have 2 copies of this one. So these can be written as lowercase, and another example of that would be like little b. Okay, so we get into even more terminology here, because we know we have dominant and recessive, but our individual combinations of dominant and recessive are either going to be called homozygous or heterozygous. So if it's homozygous, homo means same. You're going to have 2 copies of one allele. So what this looks like, it can either be homozygous dominant, where you have 2 capital letters, or homozygous recessive, where it would be 2 lowercase letters. Okay, so homozygous just meaning the same. Dominant, again, is always capital. Recessive is always lowercase. Homozygous can be either of those. Heterozygous, it's always going to be just one version. You have 1 copy of each possible allele. Usually we only look at 2 different alleles for a given trait. So an example of this would be like big T, little t. Big T is probably tallness, so little t would be short. So this is heterozygous, and you always, always, always write the dominant trait first, and that's the phenotype that is seen. So notice here I didn't write little t, big T, I wrote big T, little t. That's how you're always going to do it. And again, the recessive one is lowercase, but you won't actually see this if you're given that genotype. You will not see that phenotype. So if this was tallness, this one would be tall. You wouldn't see the short one. Okay, so just a reminder with what this looks like on actual chromosomes. So homozygous, this would be homozygous dominant. If we look at actual chromosome, you'd see like 2 big B's. Heterozygous, big B, little b, or big B, little b. But again, you always write the big one first. And then homozygous recessive would be little b, little b. Okay, so if you actually see them written on chromosomes like this, that's what they would look like. Okay, so this is just some quick examples. We're going to identify each genotype as homozygous recessive, homozygous dominant, or heterozygous. You can try to stop it and quiz yourself, or you can just follow along with me. So this first one, I have 2 big B's. Dominant is capital, so this must be homozygous dominant. Big A, little a, I've got a dominant and a recessive, so this has to be heterozygous. Another big H, little h, capital and lowercase, so this is heterozygous as well. Big H, 2 capitals, homozygous, they're the same. The capital is dominant, so homozygous dominant. Little h, they're the same, so homozygous, and those are lowercase, so it's homozygous recessive. Okay, so when you're going through and identifying, that's how you go through that process. All right, so we've said genotype and phenotype, but these two things are related to each other. So if you're given the genotype, it's going to directly affect the phenotype that you see. But if you see the phenotype, it can't always tell you the genotype because, as I've already said, if a dominant gene is there, that's what's actually going to be expressed. So recessive genes, we say they can be masked or hidden, sort of like if you wear a mask, it hides your face. So it can be there, but you wouldn't see it. So the example, big T, little t, that would make a tall plant. The short gene, the little t, is present. Okay, so at this point you might be asking, well, how am I ever going to figure out if what I'm going to see here? Well, when two people or 2 organisms of a known genotype mate together, we can infer what their offspring possibly will be, and this is called a monohybrid cross when we're just looking at one gene or one trait. And we're going to use something called a Punnett square, which I believe you've all seen before, but we're going to go through this. And Punnett square just looks like this. For one gene that we're looking at, you would just write 4 boxes. We're going to look at it with more than one trait in the future. And then you just simply write like mom on one side, dad on the other, and you break up their individual alleles, put 1 on each box. So that'd be an example of how you'd set it up. Then you just write them in, and we'll do some examples of this. So the part of this that may be a little different is the Punnett square. It's going to predict possible outcomes. And so because of this, we can express the probability of offspring that will have any of the traits, either as a percentage, fraction, or ratio. So in just a second here, we're going to go through how we do this. Okay, so I think for this it's easiest if we think of the actual fraction first, because we can use the fraction or the ratio to then go ahead and write our percent. Okay, so this Punnett square is actually showing us S, and the trait S, that's going to be, this is kind of weird, but it's going to be for the length of the second toe. So S is standing for second. A longer second toe is actually dominant to a shorter toe, so we're going to write out the probability for. Okay, so again, this is for second toe length. So if we look at this, any that have big S anything are going to have the long toe, whereas any that have only the short ones are going to have the shorter toe. So if we express these as a probability, the probability fraction ratio, the fraction of having a long toe, if we count, we've got a long one there, long one's there, long one's there, is going to be 3 out of a possible 4. If we turn that into a probability, that's 75%. So the probability that they'll have a long toe is 75%. 1 out of the 4 have 2 short ones, so there's a 25% chance that they'll have a short toe. I do want to point out here that this is a cross between 2 heterozygotes, and you always get this kind of pattern where you have a ratio of 3 of the dominant trait to 1 of the recessive. Okay, that's always going to be the pattern. So we can look at this a little bit more, but before we get there, I do want to give you a quick reminder that depending on, we break these up in the Punnett square into 2 separate boxes because during meiosis, remember those genes are going to get separated from each other, the chromosomes will. And this happens through independent assortment. And just a reminder of what that is, the separation that happens, it happens randomly. So that means that you may end up with like capital Y, little y, it could be little y, capital Y, it could be all mixed up. So the ones that are actually in there is just randomly, but it's a 50-50 chance for each allele where it'll go. That's why we write it, one in one box, one in the other. Okay, so we're going to go through 2 more examples of this. The first here, it says curly hair C is dominant to straight hair, little c. I do want to point out here that the trait is always named after pretty much usually the first letter of the dominant one. So if we have a curly-haired mother and a straight-haired father that are both homozygous for the trait to have a baby, we need to figure out the fraction that will be curly-haired, straight-haired, and ratio as well. So first thing here, we need to draw a Punnett square. And this is just for one trait. So I'm going to make a box, although it's not the best box, but it's okay. Okay, so if we do straight hair father, we can put mom over here. It doesn't really matter who we put where, but we know mom is curly haired, and it says she's homozygous, so that means she'll be capital C, capital C. I could make that capital look a little better, but capital C, capital C. All right, and then if we put dad up here, he has straight hair, and we know he's capital as well, so he's little c, because he's homozygous. Okay, so straight hair is going to be recessive here. So now if we just go in and fill it in, you always write the bigger one first if it's there. So we'll do big C, little c, big C, little c. It doesn't matter who the trait's coming from, and if there's a capital, you write it first. So in this case, we end up with all 4 of them having the same possible genotype. So all the offspring are going to have big C, little c. So this means because there is a dominant one there, the big C, all of the possible offspring are going to be curly-haired. So the fraction of curly-haired offspring would be 4 out of 4. There's not going to be any straight-haired, so it'd be 0 out of 4. Like I said, it's easiest to do the fraction first. The percentage 4 to 4, well, that's 100%, and 0% of them will be straight-haired. And then if we do a ratio curly to straight-haired, and you always do the dominant first in the ratio, it'd be 4 to 0. Okay, so we're going to look at one more here. And in humans, dimples on your face are dominant to no dimples. And if we have a homozygous recessive mother and a heterozygous father to have a baby, we want to know about if their offspring will have dimples or not. So same process here. Punnett square. Okay, so now because it's not given to us, we need to assign a letter for this trait. And again, reminder, we do it after the dominant trait. So we'll do big D and little d. Okay, no dimples will be little d. So here, the mom is homozygous recessive. So we can just put mom over here again. And then if she's homozygous recessive, that means she's going to be little d, little d. Dad is heterozygous, so he's got no option but to be big D, little d. Okay, so that's dad. So now, just fill it in. And again, you always write the big one first. So big D, little d, big D, little d, little d, little d, little d, little d. So again, you just match them up. All right, so now we're going to have some different percentages here. So if we look at these offspring, again, only the ones with the capital D something are going to be dimpled. So if we look right here and right here, we've got 2 that are dimpled. So 2 out of the 4 will have dimples. Over here, we've got 2 that only have the little d's. So 2 out of 4 are going to have no dimples. Right away, this is really easy. 2 out of 4, that's 50%. Dimpled, 50% non-dimpled. So our ratio of dimples to no dimples is 2 to 2. Okay, so with that, we are at the end of the lesson. Except we have to find out about our rabbits. I almost forgot. So with our rabbits, there are a couple of things that could happen. But you've got your white rabbit and your black rabbit. And let's say we know that the white rabbit, that's a recessive trait. So we know the white rabbit's genotype is little b, little b. But let's say that the black rabbit you originally saw at the start of the lesson was big B, little b. It was heterozygous. Well, if you fill it in, this is pretty easy to see now. This is like what we just did. Half of the offspring would be black, half would be white. So if you guessed they would be gray, you're wrong. And that means half of them, again, if we do it as a percentage or a fraction, it would be half to half, and 2 to 2 would be the ratio for them. Okay, with that, we now are at the end of the lesson. You do need to write your complete summary and complete the practice problems as well.